Right. So, just FYI, this is typo. No. So, two main things that viruses, not two, but main structural difference. Viruses are acellular. Okay. What do viruses need to replicate? They obligate intracellular parasites. Remember, obligate. Okay? Two major structural biochemical components that are absent in viruses. Ribosomes and everything that you need for ATP synthesis. Okay, not everything, but they can't synthesize ATP basically. They don't have an appropriate machinery. Okay, who discovered? I'm not gonna ask you the last name. Okay, it was plant virus, tobacco mosaic, and it was called filtrable agent because it went through the filter that contained bacteria. Does that make sense? So it was able to be filtered. Can you see the virus in light? In general, no, but there are exceptions. I'm not asking which ones, but there are. Okay? So if I ask you, you cannot see any virus. No, not that simple. Two structures found in any virus. Capsid and genome. Capsid is made of protein. Does every virus have an envelope? No. Virus is without envelope. Naked. Envelope is made of proteins and phospholipids. Modern virus taxonomy is based on genetics of the virus. What type of genome? So remember, any kind of nucleic acid. dsRNA, single-stranded RNA, bless you. Double-stranded DNA, single-stranded DNA. So that's any kind of genome, and you know genomes are based on the nucleic acid. Negative and positive. Positive, remember, positive RNA is translatable. Negative RNA has to be complementary copied into translatable. Types of capsid. Three things that you need to know. Helical, icosahedral, complex. Main parts of the bacteriophage capsid, I'm not going to rewrite them there on the slide. The capsid, the tube sheath, tail fibers, tail pins. That's it. That's all you need to know. Genomes. What I ask about types of genomes, DNA or RNA. Bacteriophage infect bacteria and archaea only. No human cells, no insects, no plants, bacteria and archaea. They do not enter the cell. Only the genome enters the cell. Main steps of the lytic bacteriophage infection. So you have attachment, penetration, biosynthesis, assembly, and egress. Okay? Egress. Now, I want to make this note. Egress equals release. So I reserve the right to use either word. You have to know that the word release or the word egress means virus leaving the cell. It has nothing to do with its nucleic acid getting out of the capsid or anything. Okay? That makes sense? Bacterial cell at the end of the lytic cycle dies. Challenges. What you need to know. Specificity. Mutations in the phage. Host. Immune response. Those are the challenges. Now, with lysogenic infection, what you have to know. In lysogenic infection, viral nucleic acid, when it enters the cell, it incorporates into the bacterial chromosome, and the cell does not die. Okay? That makes sense? I mean, it can die later, but it doesn't die right away. Okay? This is why we call it lysogenic. Phages that carry out lysogenic replication are called temperate. Prophage, that's incorporated virus, horizontal gene transfer pathway that bacteriophages mediate. You just need to know the term. We talked about it before, transduction. I'm not asking generalized or specialized. Human virus. Now this, I want to highlight, so you need to know the sequence. So for major steps, attachment, penetration, then you've got uncoding, then you've got biosynthesis, assembly, and release. So one step more than bacteriophages have. Bacteriophages do not have uncoding. Human viruses do. Okay? Attachment. What happens during attachment? Virus binds to the receptor. That's the moment when there is an interaction between the receptor and the virus. Attachment. It's not penetration, it's not uncoding, it's attachment. During penetration, virus actually enters the cell. Okay? Right? So that's attachment. Receptor is necessary for the attachment. Now, difference between permissive and susceptible. Susceptible cells have receptor. Permissive cells support replication. Virus enters the cell either via fusion or via endocytosis. Fusion is the fusion of the viral envelope, but the cell membrane. Endocytosis is the virus taken in by the endocytic pathway. One step that is not specifically listed here, remember uncoding. So uncoding is the release of viral genome from the virus itself, from the capsid. Does that make sense? It's the third step. Now, biosynthesis. RNA viruses replicate in the cytoplasm because that's where RNA is supposed to be. DNA viruses, generally speaking, replicate in the nucleus because that's where the DNA replication machinery is, and they can avoid the detection of DNA abnormal in the cytoplasm by pattern recognition receptors. So, cytoplasm, nucleus. Virus envelope membrane comes from membranes of the cell. It can be cytoplasmic, it can be Golgi, it can be endoplasmic reticulum. In general, naked virus exits leads to lysis. Can a cell survive the exit or release of envelope virus? Can? Yes, sometimes it can. Okay? So, what type of genome influenza have? Negative single-stranded RNA segmented. Enveloped. Yes, so segmented. Hemagglutinin interacts with the receptor. Neuraminidase assists with entry and release. Strains are classified based on the type of the neuraminidase and hemagglutinin. Okay, transmission routes. Respiratory. Main zoonotic reservoirs for human influenza: pigs and waterfowl. Kind of disease in birds, GI. In pigs, respiratory. So human flu causes upper respiratory tract infection, and the bird flu causes lower respiratory tract infection in humans. The answer is receptor. Remember, bird flu cannot be transmitted between humans because the receptor for the bird flu in the lower respiratory tract and transmission is not easily possible. So that's kind of the answer together. Treatment options. Just know that there is a resistance, Tamiflu, amantadine. Main problem with the treatment, resistance. Antigenic drift, slow accumulation of mutations. Does that make sense? The drift, the shift, reassortment of RNA fragments. There is an influenza vaccine. The answer why it has to be redesigned is right here because of mutations, shift and drift. Signs and symptoms, may I just skip this? You all know. Flavivirus, positive single-stranded RNA genome, enveloped. Disease, it's biphasic. Remember, what we call a flu-like disease. Flu-like first phase and second phase is either hemorrhagic fever or encephalitis. Okay? Oh, crap. Which infections cause hemorrhagic fevers? I didn't realize that I copy-pasted it and didn't correct the second statement. Which flaviviruses cause encephalitis? West Nile, Japanese encephalitis, tick-borne encephalitis. Quite obviously, you will not get a question which of the following viruses causes encephalitis, and then the right answer would be Japanese encephalitis. You know, I'm not going to ask this. Or, does this virus cause hemorrhagic fever? No, it causes encephalitis. Are they directly transmitted? Yellow fever and dengue? Yes. How? Mosquitoes. Zoonotic reservoir, monkeys. Yellow fever and dengue infections are endemic in subtropical regions all over the world. That's as far as it goes. Can West Nile virus be directly transmitted between humans? No. It's transmitted by mosquitoes from birds. US, Africa, Southeast Asia. Which flavivirus infections can be prevented by vaccinations? Yellow fever, Japanese encephalitis, tick-borne encephalitis. Main symptoms of Zika virus infection in adults: fever, retroorbital pain, rash, conjunctivitis. It's all listed there, okay? Microcephaly in fetus. Zika virus infections are endemic in Africa, tropical countries, Africa, Southeast Asia, South America. Large outbreak of mosquito-borne pathogens is not possible in the US because we have air conditioning and no marshes. Mosquitoes pretty low. Well, tell this to people who live in Louisiana. But again, air conditioning. Polio virus, positive single-stranded RNA. It's not enveloped. Reservoir, transmission route, and portals of entry exit. Reservoir, soil and water. Transmission route, fecal oral. Entry, mouth. Exit, anus. Clear, right? Poliovirus normally replicates in the gut. Neurotropic poliovirus infection is rare. 1 per 100 or 200. Neurotropic poliovirus infection affects anterior horns of the spinal cord. Symptoms: paralysis, flaccid paralysis. 2 poliovirus vaccines. What you need to know is inactivated, that is Salk, and attenuated, Sabin. Advantages of inactivated: safe, efficient. Disadvantages: provides kind of worse immune response, requires multiple boosters. So you flip advantages and disadvantages and get Sabin. So advantages: even one administration of the vaccine may be sufficient for the protective immunity. Easy to administer. It's oral vaccine. Disadvantages: may cause vaccine-derived encephalitis because reverts to the wild type. Okay. So which countries? Afghanistan, Pakistan. Wild polio. Not vaccine-derived, wild. Hepatitis C, positive, positive single-stranded RNA. Enveloped, yes. Reservoir, humans. Okay, routes for transmission. I want you to know the main routes. Like, we're not talking exotic. Blood. Accidental needle sticks, sharing needles, so on and so forth. Main challenge for researchers. That I want you to know. So main challenge for researchers that work with Hep C. No animal model. Okay. No convenient cell culture model. Does that make sense? Since there is no good animal model and no convenient cell culture model, no vaccine. We're good? So, what I really want to focus here on this one, we can really infect mice or rabbits or something like this with Hep C, and we can't take some cell type and work on it because it's convenient, so we have to have like primary human hepatocytes, which you can imagine taking human liver from a human is a bit complicated from an ethical standpoint. Symptoms, signs: cirrhosis, in a long-term perspective, liver cancer. Okay? Treatment options: you don't need to know what we call like cephosbuvir, all you need to know, there are many. So it's treatable. Can hepatitis C be eradicated? Hypothetically, yes. You treat all people with hepatitis C and cure them. Since it doesn't infect anyone but people, you can eradicate the virus. You have to spend a hefty amount of money. Norovirus. A lot of this, as you can imagine, like questions that I ask, they copy paste, so sorry for these mistakes. When I read my own writing, I don't notice a lot of typos and replacements. Just norovirus, positive single-stranded RNA virus. non-enveloped. Transmission route, fecal-oral, ID50 low. Infection mostly occurs in winter. During which stages? Any, from incubation to prodromal to disease to convalescent. Signs and symptoms, gastroenteritis. Populations that are affected the most, elderly and children, so school kids and people living in the daycare facilities. Okay? Common structural features of protozoa. No cell wall. Oh, remember, always unicellular, and they all are eukaryotes. Ectoplasm, movement feeding. Endoplasm, housing organelles. Now, trophozoites. I'm going to write it down again for you. So we're going to call trophozoite TR, okay? And cyst. What stimulates the cyst formation? I would add water or water, okay? What stimulates the formation of trophozoite? Presence of nutrients and adding water. Are we clear about this? Okay? So the formation of trophozoite is the formation of cyst is encystment. The formation of a trophozoite from a cyst is encystment. There are some typos which triggers the formation of trophozoite from a cyst. Remember another thing. Which form can feed? Trophozoite. Which form can reproduce? Trophozoite. Which form can infect? Both. Yeah. Both forms can be infectious. Trophozoite can be infectious and cyst can be infectious. We clear? 4 classes. Sarcodina, that's amoeboid motion. Mastigophora. Flagellum. Celiophora. Ciliated motion and apicomplexa. No motion. This is why they are obligate parasites. Okay, Acanthamoeba and Balamuthia. Meningoencephalitis. You don't need to know the full name. You don't need to know the granulomatous amoebic meningoencephalitis. All you need to know is meningoencephalitis. So CNS. Transmitted by broken skin disease is non-communicable. One person cannot transmit it to another. Treatable. Prognosis, I would say moderate. If treated, there's no vaccine. Entamoeba histolytica. Intestinal amoebiasis. Virulence factors are enzymes just released from the entamoeba and destroy tissue. Symptoms: diarrhea, dysentery, nausea, ulcerative colitis, stuff like that. Transmitted. Fecal-oral transmission. Amoeboma. You need to know 2 sites. Liver, lungs. Amoeba infects tissue and forms kind of a granuloma. It's important pathogen worldwide. Best preventative measures are sanitation. Plasmodium. I'm not asking about individual species. Which group of protozoa? Apicomplexa. Different species have different frequency of bouts of chills and fever. Remember that. Malaria is transmitted by mosquitoes. Tropical regions. Now, that's kind of a tricky question. Is it a zoonosis? No. No animals are involved. It's the human disease transmitted by mosquitoes. Does that make sense? Are we clear? Sorry, just disregard this. First, after mosquito bite, liver. Red blood cells are infected in the blood. Red blood cells, not white blood cells, not platelets, red blood cells. Meiosis occurs in the human. Gametocytes or gametes, doesn't really matter. Gametocytes end up in mosquito. They form zygote. Remember, definitive and intermediate host. Definitive host, sexual stage. Either formation of a zygote, like in case of Plasmodium, or if we apply it to helminths, that's going to be when two helminths have sex. Male and female adult worms have sex and start producing eggs. Okay? Intermediate host, all in between. Definitive host for malaria. Mosquito. Intermediate is human. Symptoms. You don't need to know much. So, hepatosplenomegaly and bouts of chills and fever. So, different species of different length of replication in the red blood cells. Are you with me? When the merozoites leave red blood cells, you've got symptoms. There is no vaccine against malaria, no treatable, possible control measures, controlling the vector. Protozoan causes toxoplasmosis. Toxoplasma gondii, it is apicomplexa. In healthy adults, it causes no disease. Okay? Immunocompromised are at high risk. Now, in pregnant women, it can infect fetus. Remember, it can be transmitted transplacentally. Zoonotic reservoirs for toxoplasma. Rodents, cats, cattle, pigs. Transmission routes between animals. Cats consume rodents, cattle and pigs consume cysts from the ground. From animals to humans, how we can acquire it? Three main ways. Contaminated food or drinks, consumption of undercooked beef, undercooked pork, which contains cysts, and from the cat feces when we take care of the cats. Toxoplasma gondii changes the rodent behavior, so rodents stop fearing cats, come closer, and cats eat them. Leishmaniasis, zoan for God's sakes. Leishmaniasis, Leishmania. You don't need to know species like Braziliensis or Donovani. I'm not gonna Panamensis. It belongs to Mastigophora. That you need to know. Transmission route. Scent fly. We're not in the medical entomology class, so I'm not going to ask like scent fly, deer fly, black fly. You need to know that it's vector transmitted. Three forms from the mildest to severe. Cutaneous, mucocutaneous, and visceral. Leishmaniasis, tropical, South America, Africa, Southeast India. That's it. The protozoa. Classification of helminths. What I mean, you need to know nematodes, cestodes, trematodes. Nematodes round, cestodes segmented flat, trematodes non-segmented flat. Body cavity, only nematodes, sexes, only nematodes. We clear? Main challenge in treating helminthic infestations. That's fantastic autocorrection. Anyway, main challenge in treating helminthic infestations. Helminths and humans are both eukaryotes, so it's toxicity is a problem. Work reproduction is not work reproduction. They don't go to work and then have sex at work. It's worm reproduction, okay? Because you often infected with so the problems come from an adult worm. So even if you block reproduction, adult worm still causes problems. This is why it's an effective way. Mebendazole, thiabendazole. Inhibition of microtubule synthesis. Ivermectin. Do you mind? I'm not going to write this whole like the glutamate gated. So it opens chloride channels. Chloride flows into the cells, leads to hyperpolarization of the cells and paralysis of the worm. Good? Clear? Echinococcus granulosus, cestode. Definitive, dogs. Intermediate, sheep, goat, pigs. Humans, accidentally infected. Transmission route to humans, consumption of contaminated water or food. Signs and symptoms, not really much. Liver damage. You need to know hydatid cyst in the liver. Treatment, surgical removal of the cyst. Okay? Schistosoma, whatever, Mansoni, japonicum, it's flukes or trematodes. World regions that are endemic, Africa, South America. Intermediate host, snail. Definitive host, human. Transmission route gets through the unbroken skin after swimming in the water that are infested. Make sense? Signs and symptoms of schistosomiasis. So the ones that I want you to know. Here's the thing. Rash on the skin. Okay. Bloating and diarrhea. That's the intestinal infection. Okay. Cystitis. That may long-term lead to the bladder cancer. So, complications associated with cardiovascular system. Portal and pulmonary hypertension. Renal system. Cystitis. Bladder cancer. Nervous system, CNS granulomas. Fasciola hepatica, fluke, or trematode. World regions, almost entire world is endemic. Intermediate host, snail. Definitive host, human or domestic animals. Transmission route, consumption of contaminated greens. Digestive organs, intestines, and to much larger extent gallbladder, liver. Primary food source, epithelial cells in the bile ducts. Signs and symptoms: jaundice, nausea, malabsorption, fat malabsorption, anemia. So signs of the liver damage, bile duct obstruction, stuff like that. Malignancy. So mechanism, remember, fasciola hepatica stimulates the growth of epithelial cells in the bile ducts and gallbladder. Does that make sense? Since it constantly stimulates their replication and division, they accumulate mutations that may lead to gallbladder cancer. Ascaris lumbricoides, nematode. Which world regions? South America the most, Africa, Southeast Asia to some extent. Intermediate and definitive host for Ascaris lumbricoides. Human, both. So humans are normally infected. Transmission route for human infection: consumption of embryonated eggs from the fecally contaminated food or drinks. First organ, lungs. I mean, one can argue they get in the intestine and penetrate through the walls, but I'm talking about like massive symptoms. First symptoms are respiratory, so we talk lungs. Now it coughed up to the larynx, swallowed, goes back into the intestines this way. So respiratory symptoms. Intestinal infection. Bloating, blockage, intestinal blockage, can be biliary obstruction. Possible complications: complete blockage of the alimentary canal. Strongyloides stercoralis, nematode, mainly in the South America and Africa. Worm, tropical regions. Yes, it can replicate outside the host. Intermediate and definitive hosts are humans. Transmission route. Consumption of... entrance through the unbroken skin. That's not consumption. Through the unbroken skin. Cutaneous symptoms. Larva migrants. And gastrointestinal symptoms: intermittent diarrhea and constipation and bloating. Strongyloides stercoralis superinfection is the massive amount of the worm infecting immunocompromised patients when self-inoculation. Any questions? I think I covered it pretty well.